Press the go live. Okay. So, hello everyone. Welcome back to Weekend Chat with Researchers, Season 3. As you all know, we are Bugbears, and this channel is Bugbears NCBS. And we are a research group at NCBS, headed by Dr. Ashwin, and we do some interesting stuff or that we think of interest in microbes using bioinformatics and other stuff. And this is a science outreach initiative. And you all know that it has been quite a delay since we presented any session. So this time we are kickstarting a session and a season 3. So in this new season, we have many hosts apart from me. And for those who are new to our channel, I am Nitish. And with me, there are 4 other hosts named Ganesh, Vidya, Inder, Meghna. So hi everyone from our side. So let's not delay anything. We'll kickstart the session. And so Ganesh, over to you.
Yeah, thank you, Nitesh. So before we start the session, I just have a small question to Vidya, who designed the poster. So let me just share the poster here. Just a minute. Oh. Just a minute. Sorry for the delay. Yeah. Yeah, I hope everyone is able to see the poster. So yeah, hi Vidya. So first of all, it's a nicely, cutely small, colorful poster. So I'm just fascinated by all the small things in it. So we have a small family here, very cute little family, and we have a lot of visitors. So from my knowledge in biology, I can say this guy must be an amoeba, and this guy must be a bacteriophage, and yeah, this guy is quite famous now. He must be the corona, and this rod-shaped guy must be a bacillus. And yeah, what is this? This looks like a Demogorgon from Stranger Things. And also, yeah, the title says from hostile to hostages. So yeah, who are the hostile and who are the hostages? Or can you please brief us?
It's just a bit of wordplay, really. I wanted to say that in our interactions with pathogens, if our defense is successful, then we're going to be hostile to them. But if you're not successful, then you're going to be the hostages. And it's kind of the other way around for the pathogens. And the family that you can see there is playing host to a bunch of uninvited visitors with varying levels of enthusiasm. That's basically it.
Yeah, the poster looks very interesting.
Yeah. So to understand how exactly pathogens makes hostages of a hostile host environment, we have with us Dr. Varadharajan Sundaramurthy. Varadha is a principal investigator here at National Center of Biological Sciences. His lab is interested in what entails a successful infection. His work spans over a wide variety of pathogen, ranging from malaria-causing eukaryotes such as Plasmodium, bacteria like Mycobacterium tuberculosis, to the virus responsible for today's raging pandemic, COVID. So, Dr. Varadha, we would like to begin with the question, what exactly is a pathogen? Over to you.
Yeah, thank you, Meghna. And thanks, Vidya, for that very nice poster. Thanks, Ganesh, for the and Nitish for this organizing this, Inder and Ashwin for this opportunity. Before going directly into this question of what exactly is causing the pathogens and this wordplay of hostile versus host, I thought I will just take a very zoomed out view and start from. Maybe I can share my screen.
Yeah, sure.
So as you share the screen, I would ask my my viewers to comment, use the live chat option, and feel free to ask any questions that comes to your mind. We will definitely ask that to Varadha. So please feel free to use live chat option.
Yeah, thanks, Nitish. So I thought I will start from here. This is a picture that is very famous. You guys must have heard this. It was made famous many years ago. It's called pale blue dot, and this pale blue dot you see here is the Earth, and this was a picture taken by Voyager as it was exiting the orbit of Uranus, right? And this picture recently came back into the news because NASA released a revamped view of this picture. I asked my kids who are in 8th standard and 5th standard, what does this picture mean to them? And when they see this, what are the thoughts that come to their mind? And they said that, oh, really? That's Earth? We are so small, right? And then it's also a sense of wonder of how things are and why things are the way they are. I think it's this sense of wonder and curiosity that is really keeping us going. And just I really like this also because this is a selfie of the solar system from Voyager, which was launched in the 1970s, and also gives a sense of how a little bit science works because as Voyager crossed the orbit of Uranus, there was pretty much nothing to see. Everywhere was dark, so they decided to turn off the cameras. And just before that, they said, why not we turn it around? It was facing the other way. Why not we turn it around and take a selfie of all the planets? And the result was this montage of images that you see here. And there you can see all these planets, including the Earth, as really little tiny specks. But we have this sense of space as something that is huge and vast. And in another beautiful accident, this Hubble telescope was focusing on a patch of largely empty sky, and it was in fact not a planned experiment. And somebody who was convinced that they might see something in it really had to insist on taking this particular image, and they really focused Hubble on an empty patch of sky, and what came out was something like stunning. So the sky is just teeming with millions and millions of galaxies. Each dot you see here is galaxies, right? This again tells you that how little we know really about nature, starting from the level of this galaxies. And for today's talk, we are going to go down to the opposite end of the spectrum and look at the level of microbes, right? There we live in this earth that we saw, this pale blue dot, but if we go down below many levels from where we are, there are microbes just everywhere, right? And I think in this very nice uh, talk sessions, you have had researchers from NCBS and elsewhere in the previous sessions explaining you what these microbes are, explaining the ecology and the ecosystems that they operate on, right? So we have microbes that are pretty much everywhere, all around it. We can't see them, but they manifest their presence in so many, many different ways. And today, what I will be talking to you, and as an answer to the question that was posed, is what happens when some of these microbes interact with our systems, right? Um, how do we study these microbes? One of the most common ways is to culture them, right? So in the lab, we can have specific growth conditions where we provide happy and good goodies rich media for these microbes to grow, and they will grow in the lab environment under happy conditions. Now, uh, uh, if there are any science teachers in the audience, I would really like to uh, impress upon this kind of slides. This is agar art, where in an agar plate you can just streak microbes with different colors, and you can end up producing stunning works of art like this, right? So this requires that these microbes be cultured in the lab environment. And actually, there's only a very small fraction of microbes that we know how to culture. So more vast majority of them, we don't even know how to culture them properly. So just like you have these apparently empty patch of sky teeming with galaxies about which we know pretty much nothing. Similarly, we have in our own bodies and everywhere around us, we have this world teeming with microbes, and only actually a small fraction of them is accessible for us in terms of the tools that we have to study them. Um, of all these microbes, right, the microbes that cause quote unquote diseases are still a small fraction. And actually, among those microbes, we today science has advanced enough that we know quite a lot about many of these microbes that cause diseases, particularly to human beings. Right, and there is a whole wide open question of. I hope I will have some time to touch upon that. What makes a microbe cause disease? And if we know today there are 10 different diseases that are caused by microbes, and we know which microbe causes them, we sort of have an understanding of how it causes them. We also know right now with the experience of SARS-CoV-2 that there is always going to be a 11th and the 12th microbe that is going to come, and it is going to cause a disease, right? So where is it going to come from? How is it going to cause a disease? And importantly, why was it not causing a disease in the beginning, right? I hope some of these questions we will have some time today to discuss. But I want to just take you back to this gentleman here, Robert Koch. Actually, it's an interesting science history. Robert Koch and Louis Pasteur were contemporaries, and there was also a sort of a prestige war between France and Germany for, you know, who is going to uh, take the claim, right? So Koch made a very interesting set of postulates, what is even today called as Koch postulates, and these are important for understanding the basis of diseases which are caused by microorganisms. Those were days before we had these fancy cameras, right, to capture the images from microscopes. So scientists were literally drawing the images that they saw under microscopes of those days. And here you see a set of images of microbes, which are seen as it was under the microscope. So Koch saw that there were association of microbes with diseases, and in parallel along those times, Louis Pasteur also showed that uh, uh, these tiny organisms that are invisible to eyes were actually the cause of major diseases like plague, like tuberculosis, like malaria, right? Diseases that were major killers then and even now. So there was also
Vardha,
yes.
I have a question. So,
Yes.
like, what did people think before that? Like, it seems so unimaginable to have some other hypothesis other than microbes causing diseases. So, like, can you tell us what the scene was before this came into discussion?
Yeah, absolutely. Yeah, uh, that's a very good question, and uh, we tend to go by perceptions, right? Something that we cannot see does not exist, right? And before there was an idea and understanding that the world around us is teeming with microbes, it was sort of incomprehensible to assign cause for diseases to something that we could not see. But the people, or I mean, it's also that people are inherently smart and indigenous, right? So there were a lot of these associations and correlations, right? Rats were always associated with plagues. Rats and mites and plagues was associated with plagues, right? But there was not much understanding because we could not imagine that the whole thing can be carried out by a bacteria. Malaria, for example, was always associated with swamps. Right? Although mosquitoes were there, it was not conceivable for people to think that something that you cannot see might be injected into us uh, by the mosquitoes. But to a large extent, these correlations did exist, right? And I will see if there is time, I'll touch upon these. Some of the remedies that people were using to cure these diseases before the antibiotic era was also along these lines of what could these associations meant to people also. Of course, this is a small fraction. A vast majority will be uh, similar to what we are experiencing today with SARS, right? Although there is a lot of scientific evidence, there we also see alternative views. Right? So that is a theme that continues through history. But just to pointed answer to your question will be that there were these associations, but I'm not really sure if people really were interested to make a specific causative thing to a disease, as long as there was a cure. One of the major diseases was syphilis, and it was known to be a sexually transmitted disease, right? But what exactly was the agent of transmission was not known. Yeah. Okay. So actually, this might be interesting. Something that was driving this whole thing was the fermentation industry, and when microbes were first seen under the microscope, uh, people immediately made the association between fermentation that you see in fermented drinks to the microbes that were growing in those fermentation vats. Fermentation industry was, of course, dates back to antiquity, and in every culture you have fermented beverages, fermented foods in various forms, right? But one of the first places where an association was made directly with microbes was in the fermentation industry, and that was an interesting tussle between Pasteur, Koch, and the microbiologists of that era. And in fact, this is a quote I want to read, that seeing that the wine and beer undergo profound alterations because these liquids have, because these liquids have given shelter to microscopic organisms. How can one help being obsessed by the thought that the phenomena of the same kind can and must sometimes occur in humans and animals? So I think this really is one of the basis for a theory that microbes can be causing these diseases in animals.
I have a question.
Yeah.
Like, why do they how does a normal microbe become pathogen? Like, what differentiate between a helpful and a pathogenic one?
Yes,
Like,
I'm going to come to that. I'm going to come to that, and that is a very important question, very important from many different perspectives. That will also, I think, address an earlier question that I raised, which is that today, if there are 10 diseases that we know, if there is 11th disease that is going to come tomorrow, how are we going to tackle it? And that is exactly what we are going through with respect to SARS-CoV-2, right? Was it there before? Did it not cause disease before? Did it come from somewhere? Where did it come from? I think these are very, very important questions, and I hope there is no clear one answer for this, but I hope through the rest of my talk, some snippets for these answers might emerge. Okay, it all rests on this question: what happens when what happens when these microbes interact with our cells, right? So we have to first interact with the microbes. So we are an ecosystem for the microbes, and in fact, every part of our body is teeming with microbes. I'm talking about the surfaces, right? And particularly with respect to the gut, to our oral cavity. In fact, we have more 10 times, at least estimate is 10 times more microbes than human cells in our system. So your question is, why don't all of them cause disease? Why only some of them cause disease? I will try to answer that, but before going there, I think a fundamental question that we have to gain some understanding of is what happens when our cells interact with the microbes, or in other words, in what many different ways do microbes interact with our system, right? So this slide I took from a biology lesson. Our bodies and our systems are not passive repositories for microbes. At every level you can imagine for our cells and system, we have defense mechanisms against pathogens and microbes. The first line of defense are these physical barriers like the skin, the hair, the mucous membrane. In fact, our upper airway tract is lined with these cells which are having hairs in a specific orientation, and they keep beating in an opposite orientation. Right? These thick mucus secretions trap a lot of bacteria and pathogens, which are microbes. And because of this opposite side beating, these are all constantly being sent out of the system. That is one example. And a lot of these secretions, like saliva, like tears, they are loaded with chemicals which can directly counter or fight with the microbes. So even at a surface level, we have active mechanisms that are going on at all the times to actively counter these microbes. There are also interesting examples of what is called as microbiome here. Uh, in fundamental microbiology, you would have read about different types of interactions where there is a symbiotic relationship, there is a commensal relationship, there is a parasitic relationship. So in a symbiotic relationship, you have partner A and B. It could be a microbe and it could be us, which are mutually beneficial. We have examples of that in our own gut systems. There are bacteria that we harbor that help us in digesting specific type of food. There are also evidence emerging that maybe some of those microbes can also be helping us by not letting other types of microbes, which could be pathogenic, to attack us. Okay.
Vardha, I had a
Yes.
question here.
Yeah.
Is there any chance that these commensal microorganisms, which are beneficial, they turn against us and become pathogenic? Are there instances?
Yes. So not so a lot of times, what causes a disease is also a function of where these pathogens are. And if something is in a place where it is supposed to be in nature and doing what it is supposed to do, there is a certain balance and homeostasis. And if it is not there, if it is elsewhere, it is going to result in an alteration of that homeostasis. In a lot of cases, a disease is something that is, we will see this later, that is more a reflection of how our immune system reacts to a presence of something. And there are a lot of examples where there is an overdrive of immune system. It leads to, for example, an attack of something coming from the pathogens, and which results in an inflammatory response. And when these inflammatory responses mounted by our system, that is something that can actually cause diseases. So for example, in an infection like tuberculosis, a lot of pathology is actually because of the inflammatory responses from our own immune system. So the immune system tries to be really specific to only kill the pathogen, but because an attack has to be done, there is also a lot of collateral damages happening to the system, right? And I think beyond a point, it can get quite harmful. So to directly answer your question, there are these specific strains of E. coli which are present in our intestine. And the intestinal barrier is something that is very important, right? If you think of it, the entire lining of our gastrointestinal tract is essentially very similar to the external environment, right? So if you consider the gastrointestinal tract as a tube, the inner lining of the tube is not separate from our external world, right? It continues through the oral cavity and so on, right? So the integrity of the gastrointestinal tract membrane is very important. If that is breached, then whatever is living there can gain access to our interior. The environment, like blood, for example, is a sterile environment for microbes. You don't want anything to be growing there. But whatever beneficial effects, a commensal effect or a symbiotic effect a microbe is having in the gut can only happen in the environment of the gut. If you take that microbe from there and put it somewhere else, it might cause an effect that we don't want to happen. So if there is a breach of intestine, the same E. coli strain, which is harmless or even beneficial there, can result in a disease. Maybe even not directly, but maybe because our immune system will recognize that as foreign and mount an attack there.
So most of what we call a sickness or symptom is our immune system fighting against whatever pathogen has infected us.
Yes and no. So yes, because our immune system always keeps fighting against the pathogens, and in the next slides, I'm going to show how it is being done. But a lot of pathogens also mount very specific targeted vicious attack. Okay, some of them are directly geared to counter the immune defense of the immune cells, but there are many other direct attacks which are targeted specifically for different purposes. I will mention that.
In the same line with Meghna's question, we have one question from Harini on YouTube. What does the microbe do to make us sick?
Sorry?
So the question is, what does the microbe do to make us sick?
Yes. So, yeah, that's a very good question, Harini. It does a lot of things. Okay, and when you feel sick, you feel sick in many different ways. So different microbes can do very different things to make you sick in a different way. So with something like a microbe that is sitting in your airway cells, in your lungs, for example, it will make you cough. It will irritate your system, respiratory system. There are other types of microbes that can affect your intestine, for example, and then you feel sick in your stomach, right? So diarrhea, for example, pathogens that cause diarrhea is a example of that. There are other pathogens that can attack your nervous system. There are pathogens that can cause epilepsy. There are pathogens that can cause blindness. There are pathogens that can cause muscle weakness, wasting. There are pathogens that can deform your bone. So it entirely depends on how specific an interaction a pathogen is having with the host. Okay, I'm not going to go into very specific individual examples today, but what I thought I will do is to give you a sort of broad brush strokes on how our body is geared to attack different pathogens and what kind of mechanisms pathogens have to overcome these defense pathways.
Vardha, I have a follow-up question to what Meghna asked, where there are some commensals which might become pathogens. So is there something where there's the things happen other way around, where some pathogens ultimately turn into commensals? Because in the long term, harming your own host seems like an unsustainable strategy because they also want to continue surviving, right?
Yeah, absolutely. Yeah. So I think the boundaries between what is commensal, what is pathogen is a fairly fluid one, and it is also very context dependent. Right, as I said, if things are where they are, and pathogens, microbes also, even in the example of the gastrointestinal tract in our intestine, you have a very long intestine, right? We have many meters of intestine which is coiled, and there are distinct parts of these intestine which harbor different types of microbes. If you look at the gut microbiota, right? Um, how do they know where they are? Right? There are research has showing that there are these metabolic gradients across this gut lining, where by sensing the concentration of specific metabolites, a particular microbe can know where it is, right? And maybe if that is the place, it has to be for its own optimal uh, growth, and that maintains the homeostasis of both the microbe and our system, right? Anything that disrupts this, if that microbe goes elsewhere to the gut system, for example, maybe it is not a optimal place for it, and then our body is going to react to it in a different way. Now you are turning the question other way around: is there pathogens which become commensal, right? uh, Again, I go back to my point on it's not a strict boundary. um, there's one famous example that people who think along these lines give is about mycobacterium, actually, tuberculosis, the disease that is, I think, one of the most bacteria. Mycobacterium tuberculosis is called the most successful pathogen. because it has infected so many millions of people. 1/3 of the world is supposed to be infected with the bacteria. And about 10% of the people who are infected, they actually get the disease, which means a vast majority of the people who get infected do not fall sick, right? And so the bacteria can survive in our system throughout the life of the person without even the person knowing that he or she is harboring this bacteria. Now, for that individual, right, does it really matter that there is a pathogen that is sitting inside? Is a question, a very provocative question. But there is also something that is called as a dirty secret of an immunologist, which is that uh, one of the strongest adjuvants known are mycobacterial antigens. So mycobacterial antigens are known to elicit very, very strong immune responses, to the extent that uh, one of the most commonly used laboratory reagents for biomedical research are antibodies, which are produced against a lot of different proteins. And almost all antibody production requires something from mycobacteria, because uh, to for our body to generate a robust immune response, these adjuvants can help a lot, right? So there is a school of thought which is arguing that if a vast majority of people who are infected do not show the symptom of the disease, but because they harbor this pathogen, their basal immune level goes high, can you not argue then that there is some kind of benefit that is being imparted? Right? It's a very provocative thought, very provocative question. But for an infected individual who is not showing symptom of the disease, if there is a generic increase in a immune level, then we will have to ask, is it a beneficial thing or not? And there are no easy answers for this, but I guess this kind of questions can elicit some thoughts.
Well, I'm thinking like all these things like opportunity and this like it's kind of like very selfish. Like bacteria will strike if given an opportunity. If not, then he will simply live quietly enough. Like that's very selfish, like kind of thing. Like in nature, like we say that nature is all loving and all, but no, it's very scary and selfish. Yeah, we harbor them to get antibody adjuvants, and they quietly live on us. And when we get weak, then strike us at the moment. Like it's very scary and selfish.
Yeah, we should not prescribe intent to these things that are happening in nature. A lot of time what we see is an outcome of what is happening, right? What we can do is to gain a detailed understanding of what is happening, and based on this knowledge, then we can try to have strategies to counter them. So, particularly in the context of this kind of host pathogen interactions, it is always a battle between both, right? So, uh, uh, we and the microbes are evolving together. Microbes being smaller, they can evolve faster, right? Which is why a lot of epidemics, pandemics happen. Um, but what we see is also a result of these two independent systems which are coming together. And essentially, it's a snapshot of this particular moment in time that we are seeing this effect. A lot of these pandemics are also self-limiting, right? That is another important thing to keep in mind.
I'm like still stuck on the slides that you showed before. So what happens to the pathogen when it enters our immune system? Like,
Exactly. Thank you. I'm just going to say that, right? So if you see this slide, can you see my cursor?
Yes.
Yeah. So this is a neutrophil. This was a movie made in, I think, Vanderbilt University in 1950s. This is a neutrophil, and this guy here is a bacteria, right? You just see what happens when I play this. You can see this is literally chasing the bacteria, and at some point it is going to eat that. Right? You can also see that the cell is somewhat different in this part compared to this part. This is called the leading edge of the cell. And you could see that this has actually now eaten that bacteria. In my next slide, I'll show what happens when it is eaten. But the purpose of this movie is also to tell you how strong and robust our immune cells are. They literally chase the pathogens down and eat them. Now,
Are
this is
these
a sort
are
of,
these same neutrophil cells which we get to see in our blood reports, like the
yes,
neutrophil
yes,
count?
yes, yes. So, uh, so our blood has a lot of these very special immune cells. That is neutrophil, what you saw. Now, what you see here is another type of cell called macrophage, right? Now, this is a modern version of the same movie where you will see that this thing here is actually a glass capillary, which will hold this cell in one place. Okay. And then what scientists will do is to simply show bacteria in this side. Then you see what happens. Right. Now, this cell, this macrophage is held in place by this capillary. And here they are showing the bacteria at a safe distance from the cell. Now you can see this part of the cell is generating some responses, and it is making all these protrusions, and it is trying to take that bacteria. It is not able to take because it is held in place by this capillary. Now what they will also do as the movie goes is to move this bacteria from here to here and from here to here. So you just observe the cell and see what is happening there. Right. So these immune cells are able to clearly sense where the bacteria is and definitely change their trajectories towards that and eventually gobble up these pathogens. Uh, I will show you another movie. Just see this. So there is a whole bunch of bacteria. It's a lot of bacteria here. And there is one cell here that is going to try to eat up this bacteria. Just watch what happens here. This entire chunk is now taken up by this cell, and you can see that they will slowly disappear. And now this chunk is going to be taken up and they will also disappear. You can actually see from these images that this bacteria is inside the cell now and it is slowly being eaten up by the cell. This process is called phagocytosis, and these cells are loaded with specific chemicals which is going to completely kill these bacteria. So our defense mechanisms are fairly robust and strong to mostly counter these microbes. Then why do we fall sick is going to be the question, right?
Vardha, I have this specific question. Like in last three videos that you showed, uh, the movement of these cells seemed quite directional, and it appears like that they are actually seeing the cell and they are engulfing it. So, what exactly is like directing these cells towards pathogen or towards the bacteria?
Exactly. Yeah. So that's a very good question. So there is always a sense of direction to most of these cells. They can, most of our cells. Uh, in fact, this is something that is um, through the life of our system, right? Right from the time of fertilization, uh, almost all our biology is driven by very directed movement of cells towards a particular time and space. So there are specific receptors, specific things like chemoattractants, specific mechanisms for our cells to sense its position in time and space. A lot of these infected cells will also secrete specific molecules called particular type of chemokines and cytokines, which themselves will attract other cells. So, for example, now that this cell is infected, it is going to send out signals that there is an infection happening here. Now there will be other cells. Now these are experiments that are done in cultures in cultured systems under the microscope we are seeing. But in our body, inside our body, if the cell is infected, it is going to send out signals. And there will be more such macrophages, more such neutrophils, which will respond to that signal and come to this site. Right? Now, in this site, if there are other microbes, other bacteria that are there, now those cells are going to be eaten up. So yes, the pathogens themselves are directly sensed, but the presence of these pathogens are also indirectly sensed, because these infected cells or cells which have sensed the pathogen are also going to send out signals. And as a consequence of this signal, you're going to have more immune cells which congregate to a particular place.
So they're kind of following a diffusion gradient of chemicals, right?
Yes, you could say that. Yeah. Yeah, shall I continue? So at a molecular level, how do our cells sense the presence of pathogens? And I think this goes back to a question of what is a pathogen, and how is that differentiated from what can be a normal microbe, right? Now, here is an experiment. This is a scanning EM of a fruit fly, Drosophila, right? You can see that this is the eye of the fly, this is the head, and it is intact, and it just looks good, looks healthy. Now, the thing is, this fly has been attacked by a fungus under artificial experimental condition. It has been attacked by a fungus, but this fly has fought the fungus fairly successfully. Now, there is another fly here which has been attacked by the same dose of fungus, and you can see that this fly is not able to withstand the attack of this fungus, and you can see this fungus has sort of killed the fly, and all these things are this fungus hyphae that is growing here. Right? So, what is different between this fly and this fly? So, this is a sort of experiments called genetic screens, and in clearly then they figured out there was a particular gene that was defective in this fly. Now that actually led to the discovery of this whole family of protein called toll-like receptors, or TLRs. Now these toll-like receptors, and now we have several other similar kind of family of proteins who can directly sense different chemicals. Like for example, this TLR4, toll-like receptor 4, can specifically sense something called LPS, or lipopolysaccharide. Right? Now, this lipopolysaccharide is something that is present. Where is it present? If you see, this is something that is present on the cell wall, on the outer surface of microbes, of bacteria. Now, many pathogens, mostly pathogens, harbor these kind of specific signatures. They are called as PAMPs, or pathogen associated molecular prints. Right? Now, LPS, or lipopolysaccharide, is one example. There are a variety of different such signatures. Now, these PAMPs are distinctly recognized by these kind of receptors, which are largely present in our immune cells, like neutrophils and macrophages that I showed previously. Right? So, using these specific receptors, now there is a line here. You see this yellow line. Now, this is the membrane of the cell. So, all these part of this TLR is outside the cell. So, in the right place where it can directly bind and recognize these lipopolysaccharides. Now, as a consequence of this binding, what will happen is there will be a signaling cascade initiated. So, inside the cell, there will be changes in the gene transcription machinery, and specific proteins and molecules which are important to fight these infections, in terms of, they have to degrade this, they have to send out signals that, hey, there is an infection, I have detected an infection, like those chemokines and cytokines that I told. So all those transcriptional changes will start happening as a direct consequence of these receptors, which are identifying these chemicals. Now, these chemicals, or these PAMPs, are something that is distinct for pathogens. And I think this forms the crux of where this recognition comes from. Right? Many pathogens harbor these sprints, and evolution, the co-evolution of the host and the pathogens have ensure that our systems are able to directly detect these signatures coming from the pathogens and mount a robust immune attack. And you saw in the last slide what happens as a consequence of one of these attacks.
Varadha, before we move on, we have some questions on YouTube. Could I interrupt you to ask them?
Yeah, sure.
The first one is from Harini. She said, as you said, bacteria can stay in your body forever without the person knowing it is there. So if a pathogen goes into your brain, for example, will it not cause epilepsy?
It does. Yeah. There is a worm that I forgot the name of that worm. So it causes a cystosarcosis, which can infect into the brain, and depending on where it goes into the brain, it can definitely cause epilepsy. And one of the magical thing is that if it is detected early and treated early, the worm gets killed, the epilepsy goes away. But depending on how late the detection happens, how much of the worm is there, depending on where in the brain it has harbored itself, now even if you take away the worm, the effects in terms of the neurological damage that has been sustained, they can last. Right? And that is something we have seen with cases like leprosy, for example, or even in the case of specific kinds of tuberculosis infections in the spinal cord. There are deformations that happen in the spinal cord during bone TB. So even you cure the bacteria, even you kill the bacteria, the consequences can last.
I see. And
It,
the second
it depends
question.
on where it is also, right? Just
Yeah.
to complete that question,
Sorry.
right? Brain is a very privileged, right, organ in a sense that the blood-brain barrier is very strong. It's an immune privileged site, and that is not a place where uh, anything other than our own cells, our neurons, and other cells are expected to be there, right? So the architecture is also such that there is no scope and space for anything else uh, there. So there will be very little tolerance for anything else inside a space like brain. Similarly, blood, right? Our blood is a sterile environment for pathogens. But when I said that there can be bacteria that can cohabit with us and we will not know, that largely refers to sites in our body where there is already a lot of other microbes, our airway systems, for example, right? But this is not, same will not hold for other sites of the body, brain or cerebrospinal fluid or blood or anywhere else.
So these places in our body which are known to be specifically sterile, for example, you said the brain is protected by the blood-brain barrier. What does it take to breach it? How does a pathogen, say, breach it?
In fact, a pathogen need not even reach it to cause major diseases. A well-known example can be cerebral malaria, right? Brain is a heavily vasculated organ, right? So, in fact, I think 20% of our blood flow or our oxygen consumption happens in the brain, right? So, there's a lot of these tiny capillaries that go through brain. So, even if a pathogen cannot cross the blood-brain barrier, if it can induce uh, by whatever means an obstruction in the blood flow, that can have serious effects. And that is precisely what happens with cerebral malaria, where malaria parasite is inside the red blood cells, right? But but they can modify the structure of the red blood cell, and they can produce proteins and stick them onto the surface of the red blood cell. The parasite itself is inside the red blood cell, but it can produce molecules which are on the outside of the red blood cell. Now, these can go and stick to the capillary, right? And a lot of these infected cells can congregate in the capillaries. And because these capillaries are very small and tiny, This can cause obstruction in the blood flow, and that causes cerebral malaria. But there are also this blood-brain barrier is made of tight junctions, right? These are cells which are like this. Suppose you have a pathogen, particularly eukaryotic pathogens. Now they can sort of crawl their way through these gaps. Many bacterial pathogens are also highly motile. Some of them have structures like a screw. And using this kind of apparatus, they are highly mobile. I'm going to show this in one of my subsequent slides. They can go through these tiny spaces, or they can actually drill their way through these junctions to get into the brain.
So it's
In
either
fact, malaria,
brute force or Trojan horse type of situation.
yes, yeah, you can say that, yeah.
Yeah, I'm sorry I interrupted you.
No problem.
We have
So
another
there are
question.
active mechanisms for.
I'm so sorry.
No, I
Go
was
on.
just saying what you exactly said. There are active mechanisms uh, to go where they want to go.
The second question we have from YouTube is, do pathogens develop immunity against other pathogens? How do they do it? And how is it different from a human developing immunity?
Not sure I entirely understand that, but for example, viruses, you can consider them as pathogens, right? Bacteria, you can consider them as pathogens. We also have a very human-centric view of diseases, right? Um, there is a whole uh, microbes, right? There is a whole universe of viruses which attack bacteria, and there is very beautiful, stunning immune mechanisms. You can call them immune, but there are defense mechanisms of the bacteria against viruses. In fact, the restriction endonucleases came out of such studies on bacterial defense mechanisms against viruses. CRISPR-Cas came out of such mechanisms, and quite likely that there are so many more such mechanisms which are not yet discovered. But as a general principle, if you can call that, when a virus infects the bacteria, right, the bacterial system tries to cleave or chop the DNA of the viruses, and they have to do it without damaging the DNA of the bacteria. Right? So nature has developed many mechanisms by which this can be made possible, where there is a specificity that is achieved between a bacterial DNA and a viral DNA. So this is really at the core of any immune defense mechanism, because at the heart of all of this is an ability to sense something that is not part of you, right? And if you look at this slide, all these TLRs are sensing these molecules like flagellin and CpG-DNA and NPS, and all of these are signatures which is helping our immune cells decide that I have found something that is not part of me, right? This is likely to be a pathogen. I have to attack it.
Yes.
Right. So this distinguishing between what is self and what is not self is really at the heart of all these defense mechanisms. So similarly, when a virus bacteriophage is infecting a bacteria, it is going to inject the DNA of the phage, right? So the bacterial systems have come up with very explicit mechanisms whereby they could say a piece of DNA is not from the bacteria but from the virus, and specifically launch an attack against that virus DNA. Right. So that is definitely an example of a pathogen attacking something else, which is a pathogen for itself. Right. I don't know if that answers the question, but there's also actually emerging literature on even in the case of commensals in the gut. We always think of a relationship between a particular species of bacteria in the gut with our human system. And that is also because we have a very human-centric view of the universe, right? So there are new research emerging that these gut bacteria interact with each other, right? Interact with each other and influence the environment of the gut. And what is really important or beneficial for us is not just bacteria A or bacteria B, but what is coming as an outcome of this interaction between A and B. Right, so these kind of examples are beginning to emerge now. We know very little about what is happening in this at a molecular level.
Right.
Having said that, I have a question related to the severity of infections. You also quoted about the fungal infection which infects the Drosophila, and also one common thing we know is COVID. So during the COVID, some of us were asymptomatic and some of us were symptomatic. So why do the severity of infection differ from host to host, even though it is infected by the same pathogen?
Yeah, that's a very good question. It depends a lot on the host system, right? As you mentioned the example of COVID, I'm thinking of a different example now. I will give that in a minute. We are all different, right? Our bodies are different, our genetic makeup is different, our environment is different, our food habits are different. All of this play a role in determining how our immune system responds to insult by pathogens. Now, in the laboratory, when we do infections with mice, for example, or flies, for example, we take very particular care in choosing animals of the same genetic background, which are fed the identical type of food, which are given the same kind of conditions and environment, and every possible way you can imagine. You provide exactly the same condition for all of them, and you infect them with exactly the same dose of pathogen in exactly the same way. You will still see that the responses are not identical. They will fall within a certain range, but you will see that there is a lot of difference between individuals.
I have a question
Now,
like
these differences come from the host, right? Sorry?
yeah, I have a question like you said that you take individuals for example, if there are two rats which are like identical or like are brother sister kind of thing, like they have similar genetic makeup than other. So do we also see in them different result, or they both have show the same results?
You rarely see exactly the same result.
Oh.
There will be differences, right? So where these differences come from is actually something that we are actually actively trying to address in our lab. In fact, even without going to the level of animals, if you take cultured cells in a petri dish, if you culture cells which are all identical, you still see that between two different cells the responses are different, right? Now, I will. There is actually something that I did not include in the talk today. Even the bacteria that is infecting, right? If let's say I'm infecting a group of cells with 1,000 bacteria, right, which is fairly typical thing to do in an experiment. Now there is exciting research happening, and emerging evidence is showing that these bacteria that is coming are also different. They are not exactly same in many ways. So how they are different? What are these differences? How do these differences impact the outcome of infection? These are all very exciting areas of research. But I just to go back to that example of COVID, why everybody getting COVID is not getting infected in the same way. I want to take your attention to a very unfortunate event actually that happened in Germany in I think 1920 or sometime, where kids in a particular village, they were vaccinated against tuberculosis. Right now, the vaccine strain is attenuated. That is a fundamental tenet of vaccinology. But what had happened as a result of an accident, a very unfortunate accident, was instead of providing the vaccinated strain, live virus was given to these live bacteria, live TB bacteria was given to these kids. Right, you can read this up. But it became a certain study also, in a way that as an accident, you had under controlled condition kids of very different background were given this dose of same dose of bacteria, right? What they saw was that not all these kids got the disease. Some of them got the disease, few of them unfortunately died. Some of them got the disease severity was different. Right? So that is where there was an increasing appreciation of exactly the question that you asked. Why is it that different people respond differently to diseases? Because here was an example where exactly the same dose was given as an accident to different kids, but the responses were markedly different, right? So where these differences come from, how these differences contribute to the eventual outcome of the infection and the disease, these are very important questions. These are actively ongoing areas of research, and we don't have a straightforward answer for that. Certainly, host genetics plays a big role, right? For example, you can imagine that in this case, this is a fly that has a mutation in one of these genes. So therefore, it is clearly a lot more susceptible. Right? So as humans, as an outbred population, we all have a lot of genetic variation. And so how these genetic variation contributes to disease susceptibility is also a very interesting and important area of research. No
Yes.
easy, no direct answer.
I have one question on like you stated few examples which mentioned a particular host. So is it always this case that a bacteria is dedicated to one host, or there can be multiple hosts for a bacteria or a pathogen?
Oh yes, yeah, that is also a very important question. Yes and no. So there are very specific pathogens which can infect only single species, and then on the other hand, there are these generalist pathogens that can pretty much infect almost anything. So to give an example, there is a plasmodium, right, which causes malaria. Very species specific. Plasmodium falciparum is something that will cause malaria in humans. Now, it belongs to a class of parasites called epicomplexan parasites. And in that epicomplexan family is also a parasite called Toxoplasma gondii. Now, Toxoplasma is one of the most famous generalists. It can infect any cell. Any nucleated eukaryotic cell can be infected with Toxoplasma, right? And in fact, I think almost all of us would have been infected with Toxoplasma at some point, if not now, right? So there are pathogens which adopt a very strict species-specific infection paradigm, and then there are pathogens which can be like very promiscuous. Where this promiscuity, it's also a trade-off, right? If a pathogen can just about infect anything, what is going to be the cost and what is going to be the benefit? Whereas something that is very specific, what are going to be the cost benefit? So there are evolutionary biologists who are trying to understand these trade-offs between this generalist versus specialists. But the answer to your question is both are possible.
And are there examples that these hosts play some role in some life stages of bacteria or life stages of these pathogens?
Sorry, what? I didn't understand.
So do these hosts play a role in in like life stages of these microbes, or are these hosts essential for these pathogens to survive or to continue certain life stages?
Yes, I think I'll give you an example of that. Yeah, I think that is this should answer your question. The answer is yes, because they are not infecting the host for the fun of it. They are infecting because they need the environment of the host for their survival. Now, this is a very complicated example found in nature, something that is happening all the time. And I put this slide also to take the focus away from a human-centric view of infections and diseases. This is taken from a particular issue of National Geographic, which was focusing on this infections in animals. You can see this is a frog, and you can right away see that this frog is unusual because it is having 4 legs, right? Now, this is not a genetic defect in the frog. This is not a developmental abnormality, but this is something that is actually induced by a pathogen. Right? This here is the life cycle of this pathogen. Now, what will happen to a frog that is having 4 limbs? The most obvious thing is that this frog will not be able to move around properly, right? So its motility is going to be seriously impeded. And what will happen in nature when something is seriously impeded is that it obviously gets eaten by its predator. So what is the predator for the frog? Is this bird, right? So this pathogen is making the frog into something that is going to make it as a very delicious dinner for its host. Why? Because this bird is the definitive host for this pathogen. Now, this pathogen can survive and thrive, and it is this environment of this gut of this bird is necessary for this pathogen to complete its sexual cycle, right? Then this will lay the eggs inside this bird. Now, when this bird is pooping in the water, these eggs will get released. Now, these eggs are eaten by the snails. The snails are eaten by the frog, right? And now the pathogen has to move from frogs into the bird. How? This is what it is doing to the frog. There are many such amazing examples. This is another example, incredible example, where this wasp is actually, you can see the size of this cockroach and this wasp, right? Now, this is actually made this entire cockroach a zombie to itself, right? It has injected the eggs, and this has modulated the behavior of this cockroach to an extent that this cockroach becomes a willing slave for this wasp. Now, what this wasp will do is to drag this cockroach into a safe, its own nest, so that it can grow inside this cockroach. But you see, this cockroach is much larger, right? So it will not be able to just drag it physically, but it can modulate the behavior of this cockroach to an extent that this cockroach is just going to follow this into this nest. So this is an example of neuromodulation and behavioral alterations that are induced by pathogens. This is another fascinating example. There are plenty of them. I'm just showing snippets of this. If you notice this spider, right, it is laying this web. Now, this pattern of this web is very, very unusual. Okay? This pattern of this web is not what the spider normally does. It will normally lay this very nice round webs where it can catch the prey. That is the purpose of the web. But now this pathogen that has infected this spider has modified the behavior of the spider to an extent that the shape of this web is totally changed. Now, it has made it just enough so that it can sort of hang in there so that this can grow inside the body of the spider. Now, once it grows to a certain extent, then it can just drop off. Right? So, nature is full of such examples, and I hope this answers your question on this. They need this host environment to continue their lifestyle.
Yeah.
Yeah. So in our own systems with pathogens that cause diseases, as I mentioned, similar changes happen. This is an example of a red blood cell, right? This is how it is normally. Very nice, typical concave shape. Now, this gets infected by the malarial parasite, and in a matter of few to several hours, we can start seeing extensive changes in the morphology of the cell. Again, I want to emphasize that the pathogen itself is inside the RBC. You don't see the pathogen here, but it is causing a lot of modification on the host cell. And something like this now has become something like this. In terms of for RBC, the shape plays an important role. But by altering this shape, this parasite has essentially made the RBC a bag so that it can replicate. What will happen after this is that it is going to lyse the cells and get out and infect new red blood cells. So we know actually in this example of this malarial parasite, we know a lot of details on how this is happening. The example I mentioned of cerebral malaria, where these infected cells stick to each other, is because of these kind of alterations which are induced by the parasite.
We have some questions from YouTube again. Could I quickly interrupt you to ask them?
Yeah, please.
Okay. So a question from Vinay. They are asking, is there any evidence of known pathogen which help us as a side effect of its non-lethal infection, except just immunity?
I cannot think of anything off my head, but maybe there may be examples of us trying to make use of an infection by something to counter something else. Right? Okay. I think at some point in history, there was malarial infection was considered as something that could be beneficial against something else.
Yeah.
Right? But again, we have to remember that the pathogen is not infecting us with an intent of helping us or something. It is infecting us because it needs a host. Right? And there is a variety of pathogenesis mechanisms, and there are examples of tuberculosis that I gave where we can be infected and we won't even know. There is such a fine and sustained balance between the host and the pathogen. And then you have examples like malaria here, where there is no balance here at all. Once the red blood cell is infected, his only way is for all of these events to happen, and then for the parasite to lyse the cell and get out. So there is a big amplification step that is happening here. And the goal is very clear, for the pathogen to replicate. And there are very exquisite transcriptional and gene regulatory programs that are set in place at each of these steps, so that it follows a certain set pattern there.
Yeah. We have more questions. One of them is, by taking in bacteria, does the immune cell get any extra power? If yes, in what way? If no, in why?
Sorry, I missed the first word. What was it? By what?
By taking in bacteria. So when, I guess they are asking about phagocytosis.
Taking in bacteria.
Does the immune cell get any extra power?
It's an interesting question. So by phagocytosis, do they get extra power? Actually, that is something we're trying to address in the lab. Because what is emerging is that I showed you the video of this cell that is engulfing and degrading a whole bunch of bacteria, right? Now, the cell probably is not always loaded with so much intent to actually degrade so much of bacteria. It is going to need that much of a degradative capacity, right? So the cell is probably, my guess is, not really loaded with that much capacity. But as a consequence of that infection, there's a trigger that is sent out, and some transcriptional programs are initiated so that the capacity gets built as the phagocytosis is happening. then there's some kind of a sensory mechanism that is put in place that now I have taken so much of bacteria, it has to be degraded. So we had to produce that much enzymes. So in that sense, it could be beneficial. But I think what I can also mention here is that so far I have been only talking about the innate immunity, which is these macrophages and neutrophils directly killing the bacteria. I did not talk about the adaptive immunity, which sets in a little bit later, right, which involves T cells and B cells. Right? Now, these T cells and B cells can be very specific, unlike macrophages or neutrophils, which is just going to take any bacteria that you put there. T cells and B cells are going to produce effects which are going to be specific for particular bacteria or particular viruses. Right? This is really at the heart of many vaccination strategies, where you prime your system to generate these T cells and B cells, which are going to identify specific pathogens. Right? Then these, once our immune system sees them, right, antibodies, specific antibodies will be generated by the B cells, or specific effector cytotoxic T cells are going to be produced, which are going to directly neuter these particular pathogens. But we also generate what is called as memory cells, which stay in our circulation as a certain reservoir. Now, many weeks or months or years later, if our system encounters the same pathogens, these memory cells are going to recognize them, and then they say, aha, here is an infection which I have seen before. I know what exactly to do. And then they go into a round of amplification and really mount that specific attack. So in that sense, seeing a particular bacteria can, or pathogen can does activate our immune system, adaptive immune system, in very specific ways to generate a memory response. This is exactly what is happening with vaccination, right? When kids are vaccinated, so that when in the case of the real virus or real bacteria infecting, they can generate a rapid immune response.
So Varadha, I will like to interrupt over here. So before the session, we asked like people about questions that they would want to ask in the session, and Ila on Instagram asked a particular question related to vaccination, since we are on this topic. So they asked, does multiple vaccination of one or other type has some effects on our body function? So what would you like to comment on this?
I don't entirely understand what type means, but I presume that I take it to mean, it could mean two things. I'll try to answer both. It could mean vaccines against different types of pathogens, right? And it could mean also vaccination against different strains of the same pathogen. I will try to address both. And if the questioner is asking meaning something else, maybe
I think it's in light of COVID vaccine, like
okay. Okay. Yeah. So as kids, we know that a lot of vaccines are given against different bacteria and viruses, right? And as I said, these vaccine responses are based on B cells and T cell response, which are pathogen specific. But what we do is also to mix different types of vaccines and give a combined a vaccine against a panel of diseases. Okay, so that is something that I think we are all familiar with. What happens in the case of viruses is, because they can mutate very fast, and this is in fact one of the primary challenges why we don't have a vaccine for HIV, is because HIV mutates very fast. This is a particular problem in the case of influenza, because influenza also mutate very fast. So we'll have to keep track of the mutations. And most of these immune response is mounted against the surface of the virus, right? And now the surface of this virus have very few proteins, and therefore our immune systems, the antibodies, are going to recognize these proteins. Now, I mentioned that our immune systems, the B cells, the antibodies produced are very specific, right? Now, if these mutations happen to such an extent that this antibody is not able to recognize this anymore, then we lose that specificity, which is why in the case of influenza, for every strain that comes, there is a new round of vaccination. In the Western countries, annual shots of flu vaccine is something that is fairly normal. This is exactly also what is happening with COVID. The kind of questions that are being asked is because now we see the different strains that are emerging, which started with alpha, then beta, then gamma, then delta, omicron. The vaccines were created for a particular strain. Are they going to be equally effective against a different strain? That is really the crux of what we are dealing with right now. But so far, the evidence seems to be that there was, in fact, a company that started producing Omicron-specific vaccine. And there were reports saying that this is not going to be particularly more effective than what we already have, right? So again, I think it is going to depend on the pathogen. We know from example of influenza that vaccine against the strain A is not going to be effective against some other strain. It may be effective against maybe strain B, but it may not be effective against the strain C, because we cannot really say how exactly a new strain is going to emerge and how different it is going to be from an existing strain. But there are a lot of people trying to get very predictive value into this to see how much we can predict, how fast these mutations can happen, how likely are these mutations changing the surface. Right? So there's a lot of intense theoretical and computational work that is being done in this space. So with the extent of new knowledge that has been generated at an unprecedented rate for COVID, I am pretty sure that we will get some answers to these questions.
I think that answers the question.
Yeah, I have something to ask here. So since we are talking about COVID and vaccines, have there been histories of pandemics before? And what actually happens in an infection to become a pandemic?
This is exactly my next slide. So I was just leading you there. Throughout history, if you see, sorry for this somewhat somber slide, but I thought it was important to show here. Throughout history, you can see some of the, in fact, one of the major things that have influenced human history are diseases and pandemics. And I will just briefly tell you why. This is an archaeological excavation site called Hamin Manga in northeast China. And what we saw here is a pile of skeletons of different ages and both men and women. What they noticed was that the conclusion that they draw from multiple lines of evidence was that this is 5,000 years ago. Okay? So there was a pandemic, and these were people who were all affected, and all the infected people were sort of quarantined in one place. And this is a burial site of that. So all the infected people were buried together. And this is what we are seeing as an example. You must appreciate that for something like this to have survived, there may have been many more such examples which actually did not survive, and we will not have any evidence of that. There are multiple episodes of pandemics throughout history, starting from the example I gave, and literature is in fact rife with such examples, and they manifest not necessarily as direct historical statements, but they manifest in a variety of different forms, as folklore, as music, as as in art form, etc. One of the most famous pandemic is this Black Death caused by plague in Europe. And I think it wiped out more than quarter percentage, or 25% of the Europe population. And apparently this single event has had such a lasting effect on the psyche of the people that historians say that because a large portion of people were wiped out, they were not left with enough people to do jobs, to do work, right? So cheap labor became very expensive, and historians are saying that this was something that was actually a driving force for industrialization of the society. Now there are cultural resonances of events like this, which stand to date in the form of poems and rhymes, etc. Uh, yes, so from there you can see today with the ongoing COVID how well it is being documented. Um, the Spanish influenza in the 1914 to 18 was, I think, one of the most devastating pandemics in history. I think around 500 million people, if I'm right, were affected. India was one of the most affected countries, and there are a lot of parallels between what happened at that time and what is uh, happening now with COVID. So epidemics has shaped human history, culture throughout at multiple levels. What we also know is that for all the epidemics that have, for all the pandemics that have ravaged the globe, there are many pandemics that have been localized. This great plague that the Black Death in Europe, but there are also many examples of uh, very damaging pandemics, epidemics, but which were localized. The London plague or the Marseille plague, or to give an example. Um, Even with SARS, we know that when the SARS-CoV-1 came, uh, if some of you remember, many years ago in localized to Southeast Asian region, it was localized, right, and it sort of died out, dissipated. Then the MERS came, and MERS was localized to the Middle Eastern countries, and it died out. So SARS-CoV-2, when it came, it just spread all over the world, and I think it is one of the of course, the most studied virus, but the movement of the virus was tracked to individual level at unprecedented scale. So we have very rich data that is available now to track the progress of infections across society, right? And we all know through experience how different countries and different systems have been responding to this uh, infection. So I think it goes back to the earlier question now on what makes one virus different from the other, or one pathogen different from the other. Those are different trajectories that we can take from this point on. But as a history of pandemics, I think there is a wonderful body of, wonderful in a sense of very rich body of literature that is available out there. So if there are more questions on this, I can take now, because I thought I will shift gears now to look at few other aspects. That I just put this slide to show that this was a watercolor painting of a physician who is wearing what was considered as a plague preventing costume, and I just saw a lot of parallels of that with the kind of PPEs that we are quite used to now.
No, I don't think so. There are questions on this theme. We can move forward.
Okay, fine. So what makes a bacteria, or what makes a pathogen, or what makes a microbe a pathogen, right? Um, Most of the pathogens, not all, but most of the pathogens have to infect the cells, have to get inside the cell. Right? I told about the that I don't know how I'm doing with time. Is it okay or?
I mean, it's 7:30 right now. I mean, if you feel like
Okay.
proceeding, you can.
I am okay. I'm very happy to proceed, but I don't know you guys. And
Yeah, sure. I think we can.
okay. So now just looking at it from the point of view of pathogen, right? If a pathogen manages to escape all those lines of defenses that I spoke about, uh, it reaches a cell. Now, as a pathogen, you will have to find a way to get inside the cell, right? And then you will have to find a way to, our cells also have inside the cell a lot of defense mechanisms that I spoke about. Find a way to um, uh, get rid of, to overcome all those defense mechanisms, have to find a way to divide and replicate, and find a way to get out of the cell and infect other cells, right? So that's a lot to ask. But a lot of pathogens have found very unique ways to do that, and that is beautifully exemplified in this scheme here with a pathogen called Listeria monocytogenes, which is really a nice model pathogen. You can see the cartoon here, which is infecting the cell. Now, along the periphery of this cartoon are actual data from different labs, where there is electron microscopy of showing the same process. So this is the bacteria which is being engulfed by the cell. Now, once inside the cell, most of the intracellular pathogens, they live inside this compartment called vacuoles. You can see here this bacteria inside this vacuole. What Listeria does is, once it is inside the vacuole, it will release an enzyme called listeriolysine, which is forms pores on the membrane of this vacuole and basically ruptures this vacuole. And you can see this vacuole being ruptured here, right? Now, this getting out of this vacuole is a cue. It's an important cue for this bacteria to start replicating, right? So the bacteria will start dividing only after it has crossed this stage. It's very dangerous for the bacteria to start dividing here because the environment is not conducive. That means all these transcriptional programs that are necessary for division must be activated here and not here, right? So you can see how different processes of the bacteria are tied up closely to the mechanisms of this lifestyle. Now, once inside the cytoplasm, right, cytoplasm is not a cozy place for the pathogens to live because it is loaded with a lot of things that can, our host cells can very easily detect bacteria that are there in the cytoplasm. So if the bacteria is just sitting in the cytoplasm, it is quite literally a sitting duck. So what they do is they start moving around. They start moving around very rapidly, but movement requires energy, right? And where do you get the energy? Now, these bacteria, they actually use the host actin machinery to make these beautiful actin comets. I will show you a movie subsequently. You can see now the actin machinery being recruited and these actin comets being formed. Now, using this actin comet, the bacteria will keep moving around the cell, right? Now, using this also, it can get out from one cell. It can literally, as I told in the beginning in a different example, it can literally now use this propulsion of this actin to drill its way into another cell. Right? Now, this is a neighboring cell that is infected. And if you observe closely here, in the first cell, as soon as infection, this vacuole had only one membrane. But in the second cell, it is having two membranes. Right? One membrane is coming from this cell, and this membrane is essentially coming from the nearby cell. Now, again, you can see that here. It has gone inside the neighboring cell, and it has infected the neighboring cell, and you can see these two membranes clearly here. Now, then it does the same thing. It will release this listeriolysine, and it will lyse the cell. And so this is an example of a very specific pathogen, right? Almost every pathogen does this in a very different way, in very different ways. What is common is that most intracellular pathogens will form some kind of a vacuole once they are inside the host cell. This is actually Listeria, and this is a cell that is infected with Listeria, which are these black ones. Now you can see how they are moving inside this cell. This is fascinating actually. Yeah. So we know actually quite a lot about this process, how it is happening. And I think this kind of slide should summarize a question of what is a pathogen and what makes a microbe a pathogen. Here you can see the different steps that we saw in this process. And this is in green are all the molecules of proteins from Listeria that is involved in this. And they recruit or use a lot of host proteins, which are shown in red here, to aid them in this process. And similarly, you have very different pathogens which use the host machinery in very different ways to achieve similar effects. So what makes something a pathogen will be a combination of all this, right? So these kind of proteins that are specifically produced by the pathogens are not just proteins, also lipids and other molecules. They are called virulence factors. Now, these virulence factors aid in this pathogen in one or more of these steps, which is to get inside the cell, to find a niche inside the cell, then replicate inside the cell, to get out of the cell, right? So all of this has to happen for a bacteria to be successful pathogenic. Now, as I said, each of these bacteria, they have a different and a distinct, like our fingerprints, distinct sets of these virulence factors. So the more different they are, the more such factors they have is going to influence their pathogenesis mechanisms. So some consensus answers can be on what makes something a pathogen is how in how many different ways can they manipulate a particular host cell.
Vardha, we have another question about the video from YouTube.
Mm-hmm.
What is the interval time of frames in the movie, the previous one?
I have to go and check that, but my guess is that it will be a couple of minutes, but exactly I need to go and check.
Getting back to what we were discussing right now about the various virulence factors, we have another question from YouTube by Harini. They are asking, why can few pathogens affect only few species? Does something stop them? If yes, what is that?
Yeah, good question. Very good question. Yes, something does stop them. And what stops them will be this kind of specificity in these interactions, right? They produce these specific virulence factors, which are going to interact with factors from the host, right? Now, evolution, co-evolution of the pathogen and the host has ensured that these interactions are specific at a molecular level. Now, if the host is having this molecule, which is going to have a certain shape in 3 dimensions, only a certain shape of this can interact with a certain shape of this. And these can be species specific. So even though related species can have a similar molecule, the shape can be somewhat different. Now, if this is not going to fit exactly, then it is not going to have the same kind of effect. Now, if it is not going to have the same effect, maybe the species where it is not having the same effect will be will be able to kill or deal with the pathogen more, or differently. And so that could also, if I were to turn back the question the other way around, how do new pathogens emerge? Right, I can exactly give the same answer for the same thing. So when SIV, simian immunodeficiency virus, jumped from primates into humans, there is speculation that similar kind of changes would have happened. Right. So now what happens is, for the virus, an entirely new molecules or a new field is now available to play with, something that it has not seen before. And because these viruses can mutate very fast, uh, that is one of the ideas of how HIV became very rapidly into the human population.
Is this a pattern that we see, that some pathogen, when it is able to infect a new species, it spreads very rapidly?
We
Isn't
don't
that
know actually. We don't have. I should be careful here. I'm giving this example with what is known from SIV to HIV, or we all saw how rapidly SARS-CoV-2 spread, although we don't know exactly where it has come from, right? But we don't know for every example of HIV or SARS-CoV-2 how many other such examples are there where a particular bacteria or most likely virus was able to get into human cells, but our systems was very strong and robust to completely not let them grow. That we don't know about that. We also don't know, I don't think it is likely that these jumps are one-time events, right? So maybe there is a lot of such um, back and forth going. It's a whole field called zoonosis, back and forth going between bacteria or most likely viruses across different hosts. And maybe sometimes once in a while there's a spark and then it catches fire.
Yeah, that answers the question. Thank you.
So this is an example. I just wanted to show you this. It's a very complicated slide, but just take the message I want to put from here. The inner circle here is the vacuole, is the vacuole, and inside these vacuoles are bacteria. Now, these bacteria secrete these molecules called effectors or virulence factors, right? Now, what I want to communicate here is also the gist of what causes something to be pathogenic. Now, these virulence factors can modify our system, modify our enzymes in ways which are not common or normal for us, using chemistries which are totally unique. For example, in this case, there are particular host proteins here called GDI or this GTP. You don't have to know what it is, but look at all these modifications. This what is all everything written in red here. The SID M, SID L, SID Lep T. These are all specific bacterial virulence factors or effector proteins. They are all introducing different types of modifications to this protein, right? AMPylation, CAP, phosphocholination, ubiquitination, DAMP, right? So this one bacteria, Legionella in this case, is using many different effector proteins, introducing so many different changes to this protein. Right? Now, this is RAB GDP. So RABs are really crucial proteins for trafficking pathways and many other pathways. Now, therefore, these are central for our cellular processes. So what at a cellular level most pathogens do is to identify what are all the crucial central points in our system and target them in unique ways. A lot of these chemistries involved in these modulations are unique. We don't have counterparts in our own biology, mammalian biology. A lot of these modulations are also irreversible, right? So by specifically targeting these crucial proteins, the pathogens are able to manipulate and modulate a wide variety of cellular processes. So what they do, how they do, is the subject of intense research. Yeah. Just maybe I'll stop with this slide. Pathogens need not actually all the time enter into the cell. I spoke a lot about intracellular pathogens. They need not always enter inside the cell to cause a harm. They can actually, like these cannonballs here thrown into the fort from a distance, they can produce and secrete toxins which are going to go and affect the cells. Right? Example is cholera toxin. We all know cholera causes diarrhea, right? Now the bacteria Vibrio cholerae does not really go and infect the cell, but it will secrete this cholera toxin, which then goes and makes pores, specific pores into the membranes of the cell, or similarly anthrax toxin, right? Which is why anthrax is considered as a bioterror agent, because you don't even need the live pathogen to cause a disease, but just this toxin in small amounts is necessary, is sufficient to trigger the pathology of the disease. Yeah, I think I can stop here because I have a whole section on treatment to infections. I don't know how we are doing with time. I can take more questions on bacteria, pathogens, infections, etc. And if you want, I can go through how we treat them and how these treatments have developed over time.
I think maybe it's like around 1 hour, 40 minutes, 45 minutes. We can end our session here. And I mean, it will be a pleasure if you would like to join us in other session talking about different strategies that we use to tackle these pathogens. Maybe we can start with the antibiotic era and other. And so
absolutely, I would love this because this is a lot of fun.
Amazing. So great. Yeah, we will plan that session again. And so for now, I think, yeah, we had a good amount of viewers who are still watching our session, which we planned for 1 hour. We thank you all for staying with us for this long. We thank Varadha. So thank you, Varadha, for coming us in our outreach initiative. And literally, like I really get amazed by whenever you show that video of neutrophils catching the
yes, that's a very famous video. It was made in 1950s, you know.
Yeah. And we thank different people who asked questions in the live chat box. So with this, let's end this session, and I'll say again our motto. So we believe that anybody sitting at home can learn science. You just require a small device called mobile phone and with internet connection. And that's it. And this was our first session of season 3, and we will come back in a couple or 3 weeks with another session. So stay with us, subscribe to our channel. If you are not connected to our mailing list, do join our mailing list. And with this, bye-bye.
Bye.